Hello, everyone, and welcome back. In our previous lecture, we explored the intricate structures of the biological membrane, discussing its lipid bilayer, embedded proteins, and the fluid mosaic model. Now, in this second session, we shift our focus from structure to the functions of the cell membrane. We will examine how this amazing structure actually works, especially looking at the various transport systems that allow the cell to interact with its environment. The first transport system we will discuss is ion channels. As we learned, the membrane is selectively permeable, and ions like sodium, potassium, and calcium cannot simply diffuse across the lipid bilayer on their own. They need a way to get through. Ion channels are these gateways. They are transmembrane proteins that form pore-like structures, and they are highly specific with distinct channels identified for sodium, potassium, calcium, and chloride ions. These channels are not just open holes, they are tightly controlled and have gates that open and close in response to specific signals. So, there are two main types of gated channels we need to know. Very first one is ligand-gated channels. Ligand-gated channels open when a specific chemical or ligand binds to them. A classic example is the acetylcholine receptor found on muscle cells after a nerve connection. When a neurotransmitter, acetylcholine, is released from the nerve and binds to its receptor, it triggers a conformational change that opens the channel, allowing sodium ions to rush into the muscle cells, which is the first step in muscle contraction. The second type is voltage gated channels. These channels open or close in response to changes in the electrical potential or voltage across the membrane. These are absolutely essential for the generation and propagation of nerve impulses. To summarize the key properties of ion channels, they are always composed of transmembrane proteins subunits. They are highly selective for particular ions. Ion channels are well regulated by the presence of gates, mainly the ligand and voltage types we just discussed. Their activity can be affected by certain drugs or toxins, and more importantly, mutations in the genes that encode these channel proteins can cause specific diseases, often called channelopathies. Another type of transporter is the ionophore. Unlike the fixed channels we just discussed, ionophores are small organic molecules, often produced by microorganisms. They act as mobile shuttles or carriers for ions. Their structure is fascinating. They have a hydrophilic or water-loving center that binds a specific ion, and this is surrounded by a hydrophobic or lipid-loving exterior. This allows the entire ionophore ion complex to dissolve in and physically move across the lipid bilayer, releasing the ion on the other side. Now, let's talk about water. Water is a small polar molecule, and it cannot, or it can, cross the membrane by simple diffusion. to a limited extent. However, in cells that need to move large amounts of water very quickly, such as red blood cells and the collecting ducts or collecting duct cells in your kidney, this process is dramatically enhanced by specialized water channels called aquaporins. Aquaporins are tetrameric transmembrane proteins, meaning four protein subunits come together to form a central pore that is specially selective for water molecules. To date, about 10 distinct types of aquaporins, labeled AP1 to AP10, have been identified in humans. This has important clinical implications. For example, a mutation in the gene encoding aquaporin 2 has been shown to be the cause of one type of nephrogenic diabetes, insipidus. In this condition, the kidney is unable to concentrate urine properly because water cannot be reabsorbed from the collecting duct, leading to excessive thirst and production of large amounts of dilute urine. Finally, we have gap junctions. These are not for transport between the cell and its environment, but for direct communication between adjacent cells. Certain cells develop specialized regions on their membrane that can that come into very close proximity. Gap junctions are composed of proteins called connexins. Six connexin subunits come together to form a cylindrical core or cylinder or cylindrical structures called connexin, which spans the membrane. When connexins of two adjacent cells align, they form a narrow hydrophilic channel that directly connects the systole of the two cells. This allows the passage of ions and small molecules up to about 1000 to 2000 in molecular weight, enabling rapid cell to cell signaling and coordination. To put all of this into a broader perspective, we can classify the mechanism by which substances cross the membrane into three main categories. First is passive or simple diffusion. Second one is the facilitated diffusion. And the third one is the active transport. This concludes our overview of the functions of the cell membrane, especially focusing on its transport mechanisms. We have seen how the structure we discussed in the first part enables these critical functions, from the rapid mediated by ion channels to the direct cell-to-cell communication via gap junctions. Thank you for your attention. And now I'll be happy to take any questions you may have in the comment section. If you find this helpful, please like, share, and subscribe for more lectures, and hit the bell icon to stay updated. Thank you once again.